Hi, this is Miss Linden, and this is 7th period review for our final in the spring, second semester, Unit 5. Yay! Super excited for Honors Bio. Okay, so we had 3 big topics in this unit. So we talked about cell division. When you see the word cell division, what comes to mind? Mitosis and meiosis. Exactly. So let your brain start thinking about mitosis and meiosis and the process of making more cells. Now, just right off the bat, the difference between mitosis and meiosis. What is the difference between them? Why would one do mitosis? For growth and development and repair. Growth, development, and repair. Why would you do meiosis? I heard it, sexual reproduction, exactly. So once a sperm and an egg come together, then they have to do development. And we talked about the process. Remember? The morula, right? Cleavage, 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 gastrula, right? So we'll talk through then the process of development after that. And then last but not least, then we go into chapter 23, patterns of inheritance, which is how is it that our chromosomes influence the proteins that we make and control those processes. So always go back to the big ticket items, you know, the purpose of each thing as you go through. So there's some questions here we'll work through with chapter 5, first of all, which was cell reproduction. So can you diagram the cell cycle and name the parts and what occurs? So when you think about the cell cycle, okay, there's a circle. What is the bulk of the cell cycle called? What's happening in the bulk of it? Interphase, exactly right. And then interphase can be broken down into what?
G1, S, G2.
And G2. Good. What happens during G1? Grow. Okay, and organelles? Organelles increase. Good. What happens during S? Yes, so here's your original DNA, right? It separates, and you end up having 2 strands of DNA. Dun, 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 right? And then what happens during G2? Proteins. Okay, good. And that's all interphase. And then what is this stage right here called? Yeah, it's the M stage. And you have what going on there? Mitosis. Yeah, and you can talk about, hi, you looking for something? Yeah. Oh, I might have that. Where might I have it, students? In my left pocket. Come on over. I have something for you.
Thank you.
Did you know that I love you?
Yes.
Okay, sweetheart. Have fun. I know, super cute. That's a promposal going on right now. Super cute. Okay. All right. Hi. Okay, so we have mitosis, and on top of those stages we have, do you remember what I'm starting to write right here? Cytokinesis, right? So what are the stages of mitosis? Yes, what else? Try to see if we can do them in order. Okay. Prophase, metaphase, anaphase, and telophase. Good. And cytokinesis really overlaps with that, right? Okay. So when we say, can you diagram the cell cycle, name the parts and what occurs? So when you diagram this out, you just want to have the big idea of what's going on and why. This little mitotic dance, the chromosomes right here, it's just so you can keep track of the chromosomes so you make sure each daughter cell gets an accurate copy. So all we're saying in the stages that during prophase, the chromosomes become what? Visible, and the nuclear envelope and the nucleolus disappears, radiating microtubules. They keep going. Meet in the middle at metaphase, away at anaphase, 2 nuclei at telophase, cytokinesis. Good. So when we say they meet in the middle, in mitosis they line up single file because you're trying to make an absolute clone of the parent cell. So every chromosome lines up in the middle, and those chromosomes are separated. The sister chromatids are what gets separated in your anaphase. And anaphase always has it in a way, but you're separating a copy of those chromosomes. Then telophase, what all you're doing there is you're packaging and protecting those chromosomes back up in a nuclear envelope again. And cytokinesis then is focusing on the rest of the cell. Because when you talk about mitosis, you're just talking about the nucleus and what's going on there with the chromosomes. Cytokinesis is the division of the cytoplasm. So then you would have two daughter cells that are identical. Okay, so can we diagram the cell cycle and name the parts and what occurs? We can. All right, what else is in that chapter? Could you diagram a cell and the stages of mitosis with a cell that has 3 chromosomes, 4 chromosomes, and 5 chromosomes? Do you want to do one of those?
Yes.
Okay, so we'll do 4 chromosomes. So I would start with a prophase, and a prophase usually you have the beginnings of the nuclear envelope, right? The chromosomes become what? Visible. So there's early and late prophase, but by the end of late prophase, your nuclear envelope has pretty much disappeared, and if you had, let's say, 4 chromosomes, they would all become visible. Then what stage follows prophase? Metaphase. All 4 would line up in the middle. You would have microtubules coming across. If this is an animal cell, you'd have centrioles right here, right? And then away at anaphase, so they get pulled apart. Right? And then you would go to 2 cells, extend. Then this would be branching down into 2 cells, right? Each now with 4 chromosomes in them. And you could repeat that process with any number of chromosomes. They become visible, meet in the middle, away at anaphase, 2 nuclei, right? Okay. So we're good with that one. Let's look at the next one. Can you name and describe 3 different ways meiosis contributes to genetic variety? Yes. Okay, so you are thinking about right now, I am betting, you are thinking about independent assortment. Was that what you were thinking about?
Exactly.
And so you could line up like this, or you could line up like this, right? Either way, during metaphase 1, right? Okay, what's another way besides independent assortment? Okay, number two is crossing over. So that's when they're doing synapsis like this. Let's see. You could exchange a piece of the sister chromatids, right? So you're exchanging the same kinds of information, but it could be a variation of that form, because if the green chromosome came from mom, then the red chromosome came from dad. Good job. Okay, and what is the third way? Yeah, fertilization. You don't know what sperm is going to hook up with what egg, right? Because you're going to make only 1 egg usually in females, like for us as humans, but millions of sperm. So you have no idea how they will recombine. You good with that? Can you describe the similarities and differences between mitosis and meiosis? We did that already, right? Mitosis, it's an exact what? Clone. However many chromosome numbers you have in the original cell, you will make 2 cells that have the exact same number. Whereas meiosis, you can have variety, right? And instead you will go from a diploid cell to a haploid cell. Can you tell the difference between meiosis 1 and meiosis 2? Meiosis 1, you separate the homologous pairs, and in meiosis 2, you separate the sister chromatids. When can independent assortment occur? Meiosis 1. When can crossing over occur? Meiosis 1. Yeah. So a lot of those opportunities for variety, the first 2, are going to happen in meiosis 1. Can you tell the difference between cytokinesis in plants and animals? What's something animals have? We already mentioned it once, something that animals have. Animal cells. Churros. Centrioles. Whereas plants have cell walls. Whereas animal cells do a cleavage furrow. Plant cells will build cell walls. All right, we good on that one? Okay, and can you describe the similarities and differences between oogenesis and spermatogenesis? First of all, what are you doing when you're doing oogenesis? Making eggs, and spermatogenesis, you're making sperm. Okay, so now, who's a member of the fallopian swim team? Sperm. So you make millions of them, set them loose, all going, right? So every time you do meiosis, you make 4 viable sperm, you know. But when you do meiosis with eggs, when you're doing oogenesis, you do unequal cytokinesis, so you get one large egg and a little polar body, right? And that polar body may or may not undergo meiosis too. And you do that for what purpose? Yeah, keep all the nutrients in one basket, because it's that egg that's going to support the developing zygote until it can implant in a, let's say, a uterus. Okay. Yeah? Okay. All right, chapter 22. Chapter 22 is talking about the development, and we said, can you name the structures of fertilization? So for fertilization, remember, what did we call that outer part? There was the corona radiata. I'm not sure I taught you that. Did I teach you corona radiata? No, did I teach you zona pellucida? Yes. Okay. So the zona pellucida is the jelly coat on the outside, and the sperm have to penetrate that jelly coat, right? And you have to have the sperm. Remember, what does he have on his head? Acrosome. What's in his acrosome? Enzymes to digest the zona pellucida. Just so you know, the corona radiata, you know, like there's the corona on the sun? Those are the cells outside. Those are like leftover follicle cells that came out of the ovary. Okay. So I think we hit that. Can you name and describe all the stages of embryo development? Yes, we can. We can start off. What's the number 1 stage? Zygote. Okay, that's once you have sperm plus egg, right? I'm just going to put S plus E. Okay, what's number 2? Morula. What's the morula? A solid ball of cells. Good. So it is no bigger than the original zygote, because it just does a bunch of mitosis really rapidly, and it doesn't allow for G1. What is G1? Growth. Yeah. Okay. What's number 3? Blastula. So this is a solid, and this is a hollow ball of cells. How did it get that way? Salt was pumped in. Who followed? Water, and blew it up, right? Hollow ball of cells. And then number 4 is a what? Gastrula. And a gastrula is a layered ball of cells. Remember what the layers are? What's the outermost layer? Ectoderm, meso, endoderm. What comes from ectoderm? Skin and a system. Nervous system. Good. What's endoderm? Yeah, digestive and respiratory tract and any associated organs. When we think about mesoderm, we think about muscle, bone, yeah, and everything else. That's not one of the other 2. So that's the gastrula. And then number 5 is the nerula. And this is when you start to have the development of that nervous system from the ectoderm. You have a notochord. You accept that? You need me to talk about it anymore? Okay. Can you name and describe the layers of the gastrula and what each will become? Check. Already did it. Boom. Moving on. Can you name and describe? I just said that. Can you differentiate and give an example of induction and morphogenesis? Okay. So induction is when one tissue influences another tissue to develop. For instance, the notochord, part of the mesoderm, induces the ectoderm above the notochord to invaginate in and form your spinal cord. No notochord, no spinal cord. One tissue influencing peer pressure on another tissue. That's induction. The optic vesicle induces the ectoderm to fold in and form the lens of the eye. Then the lens of the eye induces the optic vesicle to develop into the optic cup, which is your retina with your rods and cones. That's all induction. Morphogenesis is the movement of tissues during development. You know, just even forming the layered ball of cells is a form of morphogenesis. Just the changing of the structure as it develops. Your eyes start here, they move here. You know, the infolding is morphogenesis. Any actual movement or change of that structure. The beginning of organs is morphogenesis. Well, that's actually organogenesis, but it's the movement of structures in that development. Okay. Can you describe how mitosis and meiosis contribute to the stages of fertilization and development? You can. What did meiosis do for development? Made the sperm and egg. What did mitosis do in development? Everything else, right? Everything you see in development as someone grows and develops, that's all due to mitosis. Meiosis just gave you the sperm and the egg. Okay, and then chapter 23. Chapter 23, can you name Mendel's 3 laws and describe them as well as provide an example of each? So let's see if we can name Mendel's 3 laws. I'm ready. Law of dominance. Dominance. That would be the tall allele is dominant over the what? Short allele. Right? The dominant allele. Okay? What for fingers, what's dominant? 6-fingered alleles dominant over the 5-fingered alleles. What's dominant? Brown eyes or blue eyes? Now brown is the phenotype, right? But to get the brown-eyed allele is dominant over the blue-eyed allele. Okay, law of dominance. What's another one? Segregation. That means during meiosis, you're going to separate the homologous pairs, right? If there's an individual that's big T, little t, one of the gametes is going to get the little t, and one of the gametes is going to get the big T. Okay, what's the third law? Yes, independent assortment. And we already looked at where that might happen. And what that means is if you have a big T, big R, you could get big T, big R, big T, little r, little t, big R, little t, little r, right? As long as they're on different chromosomes, they can assort however they want to during metaphase 1, right, of meiosis. If they're on the same chromosome, what do we call that? Those alleles are what? Linked genes, and they won't assort independently. What's the only thing that breaks up linked genes? Crossing over. Do you remember that? Tell me if there's somewhere I left you along the wayside. If you're like, hey, Lynn. Questions? Remember, as I talk through this, highlight the part that you feel weak on. As you're taking notes, highlight it. Don't highlight the whole paper. Stop it. But things that are like, I don't remember what she just talking about. I kind of have a memory in my, past life, but highlight that and look at it so you can revisit it. Okay. Next, can you differentiate between a somatic cell and a gamete? What's a gamete? You know this. Sperm or a egg. Somatic cell is everything else. Yeah, sex cell is a gamete. Everything else is a somatic cell. Phenotype and genotype. How the genotype is displayed. Yeah, the phenotype is how the genotype is displayed. So the genotype, your actual alleles, and then the phenotype is how that looks in real life. A big B for eye color will look like brown eyes in the phenotype. But big B, little b will also look like brown eyes in the phenotype. If you want to get a phenotype of blue, you have to be what? Little b, little b. Okay? Homozygous, heterozygous. Homo is same. It could be big B, or little b, little b. Heterozygous is what? Big B, little b. Good. Blood types, multiple alleles. remember on blood types, okay? And this has to do with multiple alleles. You could be big I A, you could also have a big I B allele, or you could have a little I allele. So it wasn't just dominant or recessive. There were two things that were dominant, right? And then that leads to codominance, because codominance would be when both the A and the B allele are both expressed. Multiple alleles means I don't just have a dominant and recessive. I have some options. Another example could be you could have a T1, a T2, a T3, and a little t. That would be an example of multiple alleles. There wasn't just a dominant and a recessive. Codominance is if you got like a T2, T3 person. They're expressing both of them because they are both dominant traits. Codominance. Okay? Incomplete dominance would be like if you had a red flower having sex with a white flower and their offspring were pink. That means both alleles are expressed somewhat, right? You have a blend. It looks like blending. It's not really, but you get some red and you get some pink. Sex-linked alleles. Those were tricksy only for who? Men. Only men, because they only have one of them. Girls will have two X's, so they will work dominant and recessive traits just like they work all the other traits. No change. No change. But for guys, they only have one shot at their X. So that means if you have a recessive trait, they're going to be more likely to get that recessive trait because they only need one bad X. But if a woman is going to get it, she needs to get a bad X from both her parents. A guy will only get his bad X from his mother because what did he get from his father? The Y. Why? Because he has a penis, right? So that is going to give him the male traits. There's nothing on the X that he's going to get from his dad. Polygenic inheritance, this is like your, we talked about intelligence, height, skin color. It's not this or that, or this or that. There's all levels along the continuum because you could have A's and B's and C's and Q's and I's all contributing collaboratively to that trait. Okay, much like your group share notes. All right, questions on those terms? Yes?
So what is again the difference between multiple alleles and blood types like?
Oh, blood types are an example of where you see multiple alleles. Because it's not just like I could have A blood or B blood. I could be homozygous A, heterozygous A, I could be B, I could be AB, I could be O. That is evidence of multiple alleles. It's not just tall, short. I have a lot of different phenotypes I could have because there's a lot of different genotypes I could have. There's more variations in the alleles. I have multiple alleles. All right, can you identify and give a specific example of multiple alleles? We just did. We did this. We just did this. I have a bonus question in there. Can I leave this? We just did it. Can you do a Punnett squares with one trait and give genotypic and phenotypic results with 2 or 3 traits? One trait, if I cross 2 heterozygote tall pea plants, now if I'm going to do 2 or 3 traits, let's say I have this, I'm crossing with each other, right? Remember we do individual Punnets. I cross just the T's like I did here. Right? And then I cross just the R's and multiply those probabilities together. So if I wanted to know what are the odds, if these are the parents, of having a child that looks just like each of those parents? Well, what are the odds of getting a big T, little t combination? Half. So 1/2 for this. And then it's going to be the same because it's the same kind of cross, right? Big R, little r, cross with big R, another half. Right? Are we okay with that? So what's 1 times 1? What's 2 times 2? So 1/4 of them will turn out with that genotype. When I have more than one pair of alleles that I'm looking at, I just do individual Punnets and just multiply it all together. Not add, but multiply it together. Questions on that part? Can you explain why a test cross is necessary? If I have somebody who has blue eyes, do I need to test cross them? No. Do you know their genotype?
Yeah.
What is it? Little b, little b. But if I have somebody who has brown eyes, angry brown eyes, do I know what their genotype is? No. Because what could they be?
Big B, little b, or big B.
Yes. They could be this. So who do I cross them with? Yeah, I cross them with somebody who is little b, little b. Okay? Because I know every single time the only thing they have to give is a little b, right? So if they are this kind of brownness, if they are this, then the only thing they will give every single time is what? Big B. But if they are this, half the time they will give a big B, I would predict, and half the time they will give a little b, right? So I test an unknown genotype. I know what his phenotype is. He's got brown eyes. I just don't know how brown he is, right? So I test him with somebody who's the anti-brown, the blue eyes, and then I'll find out how brown he is. Got that? All right. Do the practice problems at the end of the chapter, you know, of the genetics. That would be the easiest way to test those. And in fact, I would do the multiple choice problems at the end of 5, 22, and 23 over the things that I asked you to know. And that'll be a good indicator of what you feel comfortable with and what you don't feel comfortable with. Go back and restudy those portions of it and then take it again. See how you do. All right? You don't have to do your additional objectives because I'm not putting them on the test. All right, I think that's it. Did I have another slide here? No. The end. And have a piece of toast if you want to. Okay, good job, guys.